The Denisovans, an ancient hominin species, were first brought to light in 2010 with the discovery of a finger bone and a tooth fragment in the Denisova Cave located in the Altai Mountains of Siberia, Russia. These skeletal remains, along with subsequent findings, unveiled a previously unknown branch of the human evolutionary tree, distinct from both modern humans and Neanderthals. Named after the cave where they were discovered, the Denisovans have since become a focal point of scientific inquiry and fascination. The discovery of Denisovan fossils in the Denisova Cave marked a pivotal moment in the study of human evolution. Excavations in this Siberian cave have yielded a treasure trove of archaeological remains, including bone fragments, teeth, and artifacts dating back tens of thousands of years. Among these finds are the original Denisovan specimens, a finger bone, Denisova, Denisova 3, and a molar, Denisova 4, which provided the furry-depalmided B. Ancient hominin group. In addition to the Denisova Cave, Denisovan fossils and artifacts have been unearthed at other sites across Asia, including the Tibetan Plateau and Southeast Asia. These discoveries have expanded our knowledge of Denisovan distribution and behavior, suggesting that they were not confined to a single geographic region, but instead inhabited diverse environments across Eurasia. Genetic analysis has played a central role in elucidating the ancestry of the Denisovans. By extracting ancient DNA from Denisovan fossils and comparing it to the genomes of modern humans and other hominin species, researchers have been able to reconstruct the genetic profile of Denisovans and infer their evolutionary relationships. One of the most significant findings from genetic studies is the revelation of interbreeding between Denisovans, Neanderthals, and modern humans. Evidence of gene flow between these hominin groups suggests a complex web of interactions and migrations throughout the Pleistocene epoch. Moreover, genetic analysis has revealed distinct Denisovan ancestry in present-day populations, particularly among indigenous groups in Oceania and East Asia. The geographic distribution of Denisovan fossils and genetic signatures offers clues about their range and dispersal patterns. While the majority of Denisovan fossils have been found in Siberia, their genetic legacy extends far beyond this region. Analysis of ancient DNA has identified Denisovan ancestry in diverse populations across Asia, from the indigenous peoples of Papua New Guinea to the Sherpa inhabitants of the Himalayas. Furthermore, recent studies have suggested the possibility of Denisovan presence in other parts of the world, such as the Americas. Although direct evidence of Denisovans outside of Asia is lacking, the discovery of Denisovan DNA in ancient Native American populations hints at the possibility of ancient migrations and interbreeding events. One of the most intriguing aspects of Denisovan research is the evidence of interbreeding between Denisovans, modern humans, and Neanderthals. Genetic studies have revealed traces of Denisovan DNA in the genomes of present-day humans, particularly in populations of East Asian and Oceanian descent. This interbreeding likely occurred as modern humans migrated out of Africa and encountered Denisovans and Neanderthals in Eurasia. The legacy of Denisovan interbreeding is evident in the genetic diversity of modern populations, with certain regions of the genome showing higher levels of Denisovan ancestry than others. Notable examples include the EPase1 gene, associated with high-altitude adaptation in Tibetans, which is believed to have been inherited from Denisovans. These genetic contributions have provided selective advantages to modern humans, enabling them to thrive in diverse environments across the globe. In addition to genetic interbreeding, Denisovan research has hinted at the possibility of cultural exchanges between Denisovans, modern humans, and Neanderthals. Archaeological evidence from Denisovan sites suggests a sophisticated tool-making tradition characterized by the production of stone tools and symbolic artifacts. These cultural innovations may have been shared among different hominin groups through contact and interaction. Furthermore, recent studies have identified genetic variants in modern humans that are thought to have originated from Denisovans and may have conferred advantages in terms of immunity and disease resistance. This genetic legacy highlights the potential for cultural and biological exchanges between ancient hominin populations and underscores the interconnectedness of human evolution. The relationship between Denisovans, modern humans, and Neanderthals was likely characterized by a complex interplay of competition and coexistence. As these hominin groups occupied overlapping geographic regions, they would have competed for resources such as food, shelter, and territory. This competition may have driven technological innovations and cultural adaptations, leading to the development of distinct tool-making traditions and survival strategies. At the same time, there is evidence to suggest that Denisovans, modern humans, and Neanderthals also engaged in cooperation and collaboration. Interbreeding events indicate periods of genetic exchange and possibly social interaction between these hominin groups. Moreover, shared genetic adaptations, such as those related to high-altitude living, suggest a degree of environmental adaptation and niche specialization that may have facilitated coexistence in diverse habitats. The genetic legacy of the Denisovans extends far beyond their ancient hominin lineage, permeating the genomes of modern humans around the world. Through interbreeding events that occurred tens of thousands of years ago, Denisovan DNA has become integrated into the genetic makeup of present-day populations, leaving a lasting imprint on our species. Genetic studies have identified numerous regions of the modern human genome that bear the hallmarks of Denisovan ancestry. These include genes associated with immune response, metabolism, and adaptation to diverse environments. Notable examples include the EPase1 gene, linked to high-altitude adaptation in Tibetan populations, and the BNC2 gene, implicated in skin pigmentation variations among Melanesians. The contributions of Denisovan DNA to the modern human genome highlight the dynamic nature of human evolution and the role of genetic exchange in shaping the genetic diversity of our species. By studying these genetic legacies, researchers gain valuable insights into the adaptive strategies and evolutionary history of ancient hominin populations. Denisovan genetic variants have provided modern humans with a diverse array of adaptations and immune responses that have contributed to our survival and success as a species. Through interbreeding, modern human populations have acquired genetic traits from Denisovans that confer advantages in specific environments and against certain pathogens. One of the most well-known examples of Denisovan inherited adaptation is the EPase1 gene, which regulates hemoglobin production and is associated with increased oxygen uptake at high altitudes. This genetic variant is prevalent in Tibetan populations and is thought to have originated from Denisovans, providing a selective advantage for individuals living in oxygen-deprived environments. In addition to adaptations to high-altitude environments, Denisovan genetic variants have been implicated in immune responses and disease resistance. Studies have identified genes involved in immune system function that show evidence of Denisovan ancestry in modern human populations. These genetic variants may confer enhanced immunity to specific pathogens or increased resistance to infectious diseases. The study of Denisovan genetic variants has provided valuable insights into human disease susceptibility and susceptibility to infectious diseases. By analyzing the genetic makeup of modern human populations, researchers can identify regions of the genome that show evidence of Denisovan ancestry and investigate their potential implications for health, and disease. For example, studies have found associations between Denisovan genetic variants and susceptibility to certain infectious diseases such as malaria and tuberculosis. Genetic variants inherited from Denisovans may confer resistance or susceptibility to these pathogens depending on environmental factors and evolutionary pressures. Furthermore, Denisovan genetic variants have been linked to a range of other health conditions, including autoimmune disorders, metabolic diseases, and neurological disorders. By understanding the genetic basis of these diseases and their relationship to Denisovan ancestry, researchers can develop targeted interventions and treatments to improve human health. The study of Denisovan artifacts provides valuable insights into the material culture and behavioral adaptations of this ancient hominin group. Excavations at Denisovan sites such as the Denisova Cave in Siberia have uncovered a diverse array of archaeological remains, including stone tools, bone implements, and symbolic artifacts. Denisovan artifacts offer clues about their technological capabilities, subsistence strategies, and social organization. Stone tools found at Denisovan sites exhibit sophisticated craftsmanship and demonstrate a high level of skill in flint knapping and tool manufacturing. These tools were likely used for hunting, butchery, and processing plant materials, reflecting the Denisovans' adaptation to their environment. In addition to stone tools, Denisovan artifacts include bone implements such as needles and awls, which suggest advanced crafting techniques and the use of animal materials for toolmaking. Symbolic artifacts, such as personal ornaments and engraved objects, provide evidence of symbolic behavior and cultural expression among Denisovans. Paleogenomics. The study of ancient DNA extracted from fossil remains has revolutionized our understanding of Denisovans and other extinct hominin species. Advances in DNA sequencing technology have enabled researchers to extract and analyze genetic material from ancient bones and teeth with unprecedented precision and accuracy. The sequencing of Denisovan genomes has provided crucial insights into their genetic makeup, evolutionary relationships, and contributions to the modern human gene pool. By comparing Denisovan genomes to those of modern humans and other hominin species, researchers can reconstruct the evolutionary history of Denisovans and infer their population dynamics, migration patterns, and genetic diversity. Furthermore, paleogenomic studies have shed light on the interactions between Denisovans, modern humans, and Neanderthals, revealing evidence of interbreeding and genetic exchange among these ancient hominin groups. Through paleogenomics, researchers continue to unravel the mysteries of human evolution and the complex tapestry of our ancestral past. Computational and analytical tools play a crucial role in the study of Denisovans, enabling researchers to analyze large data sets, model complex evolutionary processes, and infer biological and behavioral characteristics of ancient hominin populations. Bioinformatics tools facilitate the processing and analysis of ancient DNA sequences, allowing researchers to reconstruct Denisovan genomes from fragmented and degraded DNA samples. Phylogenetic methods help to infer the evolutionary relationships between Denisovans, modern humans, and other hominin species, providing insights into their divergence times and genetic affinities. Statistical methods and computational modeling techniques allow researchers to simulate population dynamics, migration patterns, and genetic admixture events, providing valuable context for interpreting paleogenomic data. Through the integration of computational and analytical tools, researchers can reconstruct the past environments, dispersal routes, and demographic histories of Denisovans and other ancient hominin populations. The Denisovans have captured the imagination of writers and authors, inspiring numerous literary works that explore their enigmatic existence and significance in human evolution. From speculative fiction to historical novels, Denisovan-themed literature offers diverse interpretations of this ancient hominin group and its interactions with modern humans and Neanderthals. Some literary works delve into the scientific discoveries and archaeological findings related to Denisovans, presenting fictionalized accounts of their lives and cultures based on the available evidence. Other works take a more imaginative approach, incorporating elements of fantasy and adventure to weave captivating narratives set in prehistoric times. Denisovans have also made their mark on the world of media and entertainment, with various documentaries, television programs, and films exploring their story and legacy. Documentaries such as The Denisovans Lost and Found and Secrets of the Denisovans delve into the scientific research and archaeological excavations that have shed light on the Denisovans' existence. Television programs and films have likewise featured Denisovans as central characters or plot elements, depicting their interactions with modern humans and Neanderthals in dramatic and visually compelling ways. These media depictions often blend scientific facts with artistic license, aiming to entertain and educate audiences about the mysteries of human evolution. The Denisovans have captured the public's fascination with their mysterious origins and ancient ancestry. From news headlines to social media discussions, Denisovan research has garnered widespread attention and interest, sparking curiosity about our distant relatives and their place in the human family tree. However, along with fascination, Denisovans have also been the subject of misconceptions and misinformation. Popular portrayals of Denisovans in media and entertainment sometimes exaggerate or distort scientific findings, leading to misunderstandings about their biology, behavior, and significance in human evolution. As such, it is important to critically evaluate and scrutinize depictions of Denisovans in popular culture, separating fact from fiction and acknowledging the limitations of our current knowledge. By fostering informed discussions and promoting scientific literacy, we can deepen our understanding of Denisovans and their role in shaping the story of humanity. The field of Denisovan research holds great promise for future discoveries that could reshape our understanding of human evolution. Continued excavations at Denisovan sites and advancements in archaeological techniques may uncover new fossils, artifacts, and evidence of Denisovan behavior, shedding light on their biology, culture, and interactions with other hominin groups. Furthermore, ongoing paleogenomic studies are likely to yield additional insights into Denisovan genetics, population dynamics, and evolutionary relationships. Improved DNA sequencing technologies and analytical methods will enhance our ability to extract and analyze ancient DNA from fragmented and degraded remains, allowing for more comprehensive reconstructions of Denisovan genomes and their contributions to the modern human gene pool. Technological innovations in fields such as paleogenomics, archaeology, and anthropology will play a crucial role in advancing Denisovan research in the coming years. Developments in DNA sequencing, ancient protein analysis, and isotopic dating techniques will enable researchers to extract more information from ancient fossils and artifacts, providing new avenues for exploring Denisovan biology and behavior. Advances in computational and analytical tools will also facilitate the integration of diverse datasets and the modeling of complex evolutionary processes. Machine learning algorithms and statistical methods will help researchers uncover hidden patterns in genetic data, reconstruct past environments, and simulate population dynamics, enhancing our understanding of Denisovan ecology and adaptation. As Denisovan research continues to evolve, ethical and legal considerations will become increasingly important. Issues surrounding the ownership, preservation, and access to archaeological sites and genetic data must be addressed in a responsible and equitable manner, respecting the rights and interests of indigenous communities, researchers, and the public. Furthermore, ethical guidelines for the study of ancient human remains and genetic material should be established to ensure that research is conducted ethically and with sensitivity to cultural and religious beliefs. Collaboration with local communities and stakeholders will be essential for fostering mutual respect, trust, and cooperation in Denisovan research endeavors. The study of Denisovans has unveiled a wealth of information about our ancient relatives and their place in the human evolutionary tree. From their discovery in the Denisova Cave to their genetic legacy in present-day populations, Denisovans have captivated the imagination of scientists and the public alike. Denisovan research has profound implications for our understanding of human evolution, migration, and diversity. By unraveling the mysteries of Denisovan genetics, behavior, and interactions with other hominin groups, researchers gain valuable insights into the complex tapestry of our evolutionary past. As we look to the future of Denisovan research, we are filled with anticipation and excitement for the discoveries that lie ahead. By harnessing the power of technology, collaboration, and ethical stewardship, we can continue to unravel the mysteries of the Denisovans and unlock the secrets of our shared human heritage. In doing so, we honor the legacy of our ancient relatives and reaffirm our commitment to understanding the story of humanity.